Hello, everyone, and welcome back to the second part of the recording on chapters 11 and 13. We left off in the last part of the recording with this overview of the domains, the prokaryotic domains. And we mostly ignore archaea and the parts of the domain bacteria that don't cause diseases, and we focus in on the gram-positive and the gram-negative bacteria that do cause diseases in humans. That was the end of part one. Moving now into part two, we're going to focus on things that are even smaller than bacteria. Remember, the bacteria weren't even discovered until the 1800s because the microscopes weren't powerful enough. Now we're going to look at some things that are even smaller than bacteria and took even longer to discover. And that is moving into chapter 13. And we're going to focus on viruses for the bulk of it. And then at the end, we're going to focus on these structures referred to as prions, or you might hear them called prions. It's the same thing. These are even smaller than viruses. The prion, where this comes from, is PR for protein, and the IN is for infectious proteins. I don't know how to describe it other than they influence the shape of other proteins that are similar to them. It's crazy. Dude who described it won a Nobel Prize for this because it was so new, so different from so different from prokaryotic organisms, even different from viruses. There's not a cure for the prion diseases yet, but we're going to spend a little bit of time on it just because it's so interesting. Okay, so to get an idea of scale, here is a red blood cell, a cartoon of a red blood cell, and then this rod-shaped thing is representing a bacillus bacterium. And the things that look like crumbs down here, that's what we're going to be talking about. So if you zoom in on the bacterium and make it this big, 750 nanometers is the scale here, now we start to make out, oh, there are some things that are smaller than that. Let's zoom in even more, and we can see here are individual viruses of different types. Viruses, it is bizarro world. They're just so different from all of the cellular organisms. These things are not even one cell big. And there's this debate about whether we should call them alive or not alive. And I'll tell you why. It's nothing really philosophical. It all comes down to how you define life. But we'll look at that. I have a link to a website. This link, it has a diagram kind of like this. There's some 12-point font for comparison, and then a coffee bean and a sesame seed, and you zoom in and further, and there's a slider bar. Anyway, if you want to go to that, it's linked in the PowerPoint. And you can see, like you have to zoom really far in before you even start to detect viruses. Okay, so some of the characteristics of viruses. First of all, they're acellular. Once again, we are not talking about even bacterium. So these things, they do not have membrane-bounded organelles, neither do bacteria, but man, they just don't have a lot of the other structures that cells have. Here's the thing that's curious. They are obligate parasites. Obligate means it's necessary, like they can't make copies of themselves unless they are inside a host cell. And this is where that definition of is it alive or is it not alive, this is why some people would say we do not call these things alive, because they cannot do anything for themselves. Okay, so here's another weirdo thing. So all cellular life, bacteria, archaea, eukarya, every last blessed one of them has double-stranded DNA as the genetic information. And we thought that's the key, that's all of life. Well, viruses, some of them use DNA like we do, but some of them use RNA as their genetic information. That's all they carry along with them. Think about this. So that remember, in us, the DNA is double-stranded and the RNA is generally single-stranded. Imagine an obligate parasite. This thing, it travels light. So if you can get away with a single-stranded molecule instead of a double-stranded molecule, that's half the weight. And so it's less to get around. Think of this like somebody coming to your house as a guest. They come in and they're like, oh, hi, nice to see you. I didn't bring anything. So I'm just going to use your stuff. So in the kitchen, I'm going to use your pans and the food that's here in the pantry. That's cool. And then the toothbrush. Yeah, I didn't have a toothbrush. I'm going to use your toothbrush. Hope that's okay. Your wife, man, she's really cute. Gonna use that too. So like these things do not carry stuff with them. They're just going to take advantage of our stuff that's in our cells. And I'm trying to drive this point home with this genetic material thing. Like they carry so little that they make us make copies of their stuff for them. So yeah, so every last place where they can make copies of themselves, there's no like good relationship. They cause infections in other things. Certain viruses target certain bacteria, certain viruses target uh, plants, some of them target animals. Yeah, crazy. But all of them have to be inside one of those other living cells in order to make copies of themselves. And then this is where this debate about whether it's alive or not. Again, I'm not going to get caught up in that because it's nonsense. It's just what it comes down to is how do you define life? And if you define life as carrying out metabolic pathways, that this is not alive because it has to depend on us for all of our machinery, or I say us, I mean all cellular living things. They can't grow. It doesn't, they get produced inside of our cells, but a virus, once it's released, it's not like it gets bigger. And it can't reproduce independently. And so if these are your definitions for living, then viruses are not alive. So they recruit the host cell's metabolic pathways in order to reproduce themselves. And this is what I was saying, like I'm gonna come in and use your food and use your toothbrush and what did I say, girlfriend, wife, children, I don't know, whatever. I'm gonna just use all your stuff. And there's no cytoplasmic membrane, that cell membrane that all other, like all cellular life has. They don't have that. Some viruses kind of steal part of the cellular membrane as they're leaving. They kind of take it as a cloak around themselves. But they don't have organelles. They don't take the organelles with them either. They just use them while they're in our cells. And they exist inside, outside of the cell, as they're traveling from, to find another host cell, and they can also exist inside of our cells. Okay, outside of our cells, this extracellular state. I don't usually use this word, but you'll see sometimes the word virion. Notice this is different from something called viroid that we'll mention briefly later. I'm not going to make a big deal about it. I just want you to see the word in case you come across it later. All it means, it's the virus that's not actually inside of a host cell at the moment. That genetic material, I told you it could be DNA or it could be RNA, but it's always protected around the outside with this structure that's made of proteins. And again, the virus gets us to make its proteins for it. It's around the genetic information. Another term I'm not going to use. Like I said, some of the viruses have a phospholipid envelope, and that's something that they've stolen on their way out. And this outer layer provides the virus with some protection, but it also provides it has some proteins on the outside that we made for it, that it uses to dock on the next host cell, get into the next host cell. And then inside our cells, or inside whatever host cell the virus is using, that protein coat, that capsid, that gets removed. And then the virus exists basically as nucleic acid. It's just like this short segment of either DNA or RNA, a nucleic acid sequence. Crazy. So here's a cartoon on the left showing nucleic acid. This would be the viral genome. In this case, it looks like it's double-stranded DNA. And then there's some protein coat. Usually it's the same protein repeated again and again. And again, look, do you see ribosomes? No, this thing cannot make its own protein. So that's why it has to come into us, or at least inject this genetic information into us, host cells, so that we, the host cells, will make copies of not only the genetic information, but then also do the transcription, do the translation, make a bunch of these proteins for it, and then send it on its way to infect other cells. I mean, I'm not happy about viruses because, of course, they're parasites on us, but oh my gosh, as far as an evolutionary strategy, that is genius. Don't carry any of my own machinery, don't need to make ATP, just steal stuff from everybody else. This is a scanning electron micrograph of basically strands of DNA. So this is a partial genome of Escherichia coli. This is a bacterium, and we know they have less genetic information than eukaryotic cells, but look at this, the viral genome is just this little bit of DNA over here. Very, very small. And so on this genome would be the instructions for making what? The proteins that you see over here. Ah, genius. So this is where it gets tricky. The genetic material for viruses, there's more variation here than there is in any other form of life. Of all of them put together, all of the different living things, all bacteria, archaea, and eukarya, all of them double-stranded DNA. This, remember when we were talking about how do you classify the bacteria, you know, because you don't have bird wings, you don't have beaks, you don't have all these other structures to compare. So what do we have in common? We have ribosomes in common. So let's compare the ribosomal RNA, the sequence of that, and that's how we're going to do this. These things don't even have ribosomes, so you can't even compare those, for crying out loud. So instead, what scientists do is categorize the viruses based on what kind of genetic information are they carrying? Is it DNA? Is it RNA? Is it double-stranded or is it single-stranded? A particular virus will use either DNA or RNA, but not both. And the DS and the SS stands for double-stranded or single-stranded. And again, more bizarro world. Could be DNA that's double-stranded like we have, but there's some that have single-stranded DNA, some that have double-stranded RNA. What? Some have single-stranded RNA. Sometimes the genetic information can be linear and there are sections of it, or it could be one single bit of nucleic acid and it's a circle, you know, it connects back onto itself. The genomes are much smaller than the genomes of cells. Well, think about it. There's not that much to code for because this thing's not carrying a lot with it. And so just to have an idea, here are the different families of viruses that affect humans. And some of them carry DNA as their genetic information. For example, some of the herpes viruses, they're double-stranded DNA, papilloma viruses. And then we also have some that carry, there's RNA. And then some of it's single-stranded, some of it is double-stranded. All right, here's a bit of information that's important. There are some viruses, you've heard of them before, they're called retroviruses, like HIV, for example, is a retrovirus. And what that means, that retro means something's going backwards, and it's this. The virus carries RNA, but the thing still goes through the central dogma, DNA to RNA to protein. And so what it does is with this RNA, it has to make DNA off of that template. So you're kind of working backwards. And that's, well, we'll get back to that. So the HIV virus submits, injects, gets the RNA into the cells, and then the cells have to, from that RNA, kind of work backwards to make the DNA, requiring something called, well, it's a DNA polymerase, but it's retro, it's working in reverse. Human cells never have to go from RNA to make DNA. If we're going to make DNA, we're just copying DNA from DNA. And so this is one of the few targets that we get for a medication that acts against some of these retroviruses, is affecting the enzyme that does that process, because our cells never need that. So any cell that's being affected, it's being affected because there's an infection with an HIV. I'm not sure if that made any sense. Let's go on. So hosts of viruses. Now viruses, they're not generally generalists. They can't just go into any old cell. They typically only infect a particular species. And then on top of that, they tend to affect only the particular cells of that species. Or if they affect more than one species, they still only go into one kind of cell from those different species, those different hosts. And what it comes down to is the shape of the proteins that the virus has on the outside. I think I may have used this analogy before, talking about how viruses get into cells. And I think I told you about thinking in terms of robbing a house or breaking into a house. People who are going to burglarize a house, they don't bust holes in walls. They basically come in through a door or a window, some weakness that's already there. Imagine that it's only going to come into a particular kind of window. The Pella windows are supposed to be really good, but if you have a key that opens Pella windows, then you might be able to get into multiple houses using that same key. And that's what viruses do, is they exploit a particular weakness, but they can do that same weakness in different cells. That's what I'm talking about here. Their connection is so specific, they can only infect a particular kind of cell. Some bacteria sorry, not bacteria, some viruses are more generalist, but again, it comes down to what's the key. If lots of different cells have this kind of a lock on them, then those are the cells that virus can get into. You have probably heard that there can be infections that live in the body or stay in the cells for decades. They just live on. When that happens, we say that the virus is dormant in the host cells. We call them latent viruses. And when that happens, what's really going on is that the genetic information of the virus has been kind of slipped into our general DNA for that cell. Weird, but true. And they can be in there for years with no apparent viral activity. Think chicken pox. Somebody can have a chicken pox infection as a child, and then years, decades later, they can have shingles, which is like a different disease. It appears to be a different disease, and it shows up because the DNA for the chicken pox actually get spliced into some neural cells, and then years later get activated. Some latent viruses do not become incorporated into the host chromosomes. But if they do become incorporated, that incorporation, that's permanent. That's just in there. Now you might say, well, why doesn't that get passed on to the offspring? Because it gets incorporated into somatic cells, not into the cells that produce sperm or egg. That's why that doesn't get passed along. But as evidence that the virus's genetic information got like put into the DNA of our cells is that it may impact some of the proteins that are helping to control the cell cycle. And it's just like bad editing job, you know, like put in the genetic information and they just happen to put it in a bad space that's going to lead to problems of that cell controlling its cell cycle years later, possibly. And so here, an example of this is a human papilloma virus, the HPV virus. Lots and lots of folks have kind of long-standing HPV infections, and may or may not have genital warts to show for it, but may develop cancer later in life, like the cervical cells, the cells of the uterus, the cervix of the uterus may become dysplastic, may start making too many copies of themselves later. And so this is one of the big advantages for the HPV vaccine, is that even if that virus comes in, our immune system can detect it and take care of it before that DNA gets incorporated and then can cause problems down the road. And we'll finish on this point. Are viruses alive? Again, it's not a deep philosophical question. It all comes down to how do you define life? So some people say these are not living things. They're just complex chemicals, you know, a collection of chemicals that under the right circumstances can make copies, but they can't do it independently. So we don't call it being alive. Some folks will define them as being they're living, but they're just the least complex. They have the fewest parts to them. And they use these sophisticated methods, the keys to get into particular locks. They have the ability to take control of their host cell, which sounds really diabolical when really all it is our cells are in some ways kind of dumb. Like you give them some RNA, they know what to do with that. They'll start transcribing that. They'll start translating that and the ribosomes can read that messenger RNA and start making the proteins that they're being told to make. They don't check credentials or anything. And then once this genetic information from the virus has gotten into a living host cell, it causes the host to make copies of them. Oops. And so again, in this class, we're not going to make a big fuss about it. It just comes down to your definition of what is living. And if your definition of living is simply can make copies of themselves, then these things are alive. If your definition of living requires that they be able to produce metabolic reactions independently, then no, they're not alive. All right, and we will end the viruses there. In the next section, we will go on to talk about these structures called prions. And I say here, end part 3, so I must have put 2 things together. Hope you enjoyed. See you over at the end section.